right, folks, we'll let Raymond here take things over about his crayfish research with ESF.
Okay. Let me show you how this works. Okay, so. Alright, hello everyone, my name is Raymond Bailey. I'm a SUNY ESF undergraduate student in Dr. Christopher Whipps' lab. And today I'm going to be talking to you guys about the impacts of abiotic and biotic factors on crayfish worm diversity and abundance on the big water crayfish, which is basically a niche topic within a niche topic. But before we jump into the symbiont of the crayfish, we have to understand a little bit about the crayfish themselves. And so something that a lot of people don't know is that crayfish are incredibly diverse. 60 to 70% of the crayfish diversity worldwide is within the United States alone. And unfortunately, 32% of all the world's diversity is still threatened with extinction because no one seems to care about invertebrates. There's a kind of a recurring theme around this conference, it seems, or at least people have been saying. And then another 21% of them are data deficient, so we don't even know if they're endangered or should be endangered or not. And then within the United States, we only have 6 federally listed endangered crayfish species, despite 65 plus of them being narrow-range endemics, some of them only living in like a handful of places within the same county. So a lot of understanding the ecology and diversity and conserving basically the entire crayfish taxa falls to U.S. managers. So I always just kind of like to point that out. And then, of course, out of all the colorful ones, I picked the brown one. So the big water crayfish is a larger crayfish that's native to New York. It typically has a lot more of the symbionts than other crayfish that I've seen, and it's fairly tolerant. So that's why I decided to pick this, because I could find it basically everywhere. But what about the symbionts themselves? Oh, sorry, I lied. So crayfish as a whole are polytrophic ecosystem engineers. They function as predators, they function as prey, they function as scavengers. They can modify substrate structure in macroinvertebrate communities. They serve as efficient modes of energy transfer across trophic levels, which includes being a reciprocal subsidy to terrestrial systems because of ducks and birds, snakes, otters, etc., raccoons. And just to put into perspective for anyone who's a DC manager, 50 to 70% of smallmouth bass diets in the summer and fall are almost just crayfish, purely just crayfish. And in some systems, they can be up to 50% of the total benthic invertebrate biomass. So they make up this big chunk of the ecosystem's biomass. And they have been negatively correlated with smaller centrarchid species, darters, and even amphibians. So suffice to say, they are kind of like this hidden cog in ecosystem machinery that goes overlooked a lot of the time. And one of the parts that's overlooked about them is their symbionts. So unless otherwise noted, all these photos are mine, because I can't find any online of the symbiont, so I had to dig more. So these guys are Branchiodellida, or crayfish worms. They are a sister group to leeches. They used to be considered leeches, but have recently been split off. They're obligate ectosymbionts. They live on the outside of the crayfish exoskeleton, and they're obligates because they need a crayfish to reproduce on, but they can be free-living. They feed on exoskeleton microbiome like rotifers, detritus, algae, diatoms, that mix of things, as well as not even potentially that it could be true, but potentially they could feed on gill tissue as well. They are able to spatially distribute themselves around the crayfish. So, for example, the Xeronogotin instabilis, which I refer to as the tennis rackets, because they look like tennis rackets, and Latin is a mouthful, and they have no common names. So the tennis rackets, they're only really found in the claws, while some are only found in the gills, and some are only found on the abdomen and legs and stuff. And then they also dietarily partition themselves, so some are eating more rotifers, some are eating more algae, diatoms. It just kind of depends on the species that you're looking at. And they also would impact crayfish behavior. So for example, invasive signal crayfish have been known to reduce foraging and aggressiveness with Branchiodellid communities. And so that has obvious impacts for how invasion fronts are changing. Like rusty crayfish don't seem to have many symbionts at all, and they seem to be disrupting the abundance of Branchiodellid's on native crayfish. So there might be some interplay with disrupting a symbiosis with their invasion front as well. And more importantly, they can be in high and low densities. So they could be up to 1,800 different crayfish worms on a single crayfish of 5 different genera and 8 species. So it's like a little microbiome within a microbiome, basically. But yeah. So these are supposedly the species in New York. As Owen was saying, think yesterday, the last time crayfish were really surveyed in New York was like 1950s or so. These guys are similar. Well, all these records come from like 1952. So maybe they're in New York, maybe they're not, which is kind of one of the goals of the study is to see what species are still here. Camerocola philadelphicus has question marks next to it because it is a species complex, and as you will see soon, it shows up in many different spots in the phylogenetic tree with many different genera. So that when I say Camerocola philadelphicus, I mean one type of that one. So yeah. But what actually drives their abundance and diversity on individual crayfish? So I was thinking that worm abundance changes with increasing temperatures of pH, and when I reference the literature, I mean basically 5 papers, because there's not much on how abiotic stuff impacts these guys at all. So a lot of this is very descriptive. And so, yes, the null hypothesis is that one study in Iran, which did the only temperature thing that I could find on these guys, showed that crayfish worm abundance increased with increasing temperature. Obviously, that's halfway across the world, so we're going to see if that holds true here. And then worm abundance is greater in larger male crayfish, which I could explain more later. Worm diversity is greater in crayfish diverse sites because these worms can be species specific, but as crayfish species are constantly being split and worm species are constantly being reformed, like Camerocola philadelphicus was renamed like 3 times in the past like 20 years or so, which makes finding papers on them ridiculously hard. So hopefully in more crayfish diverse sites we'll have more worm species, and then the overall arching goal is to find all the branch-bedelid species listed in 52 for genetics, because a lot of them haven't had genetics done on them before. And this is kind of like 2 quotes from one of the only papers that has done this. So no studies have empirically explored the effects of water chemistry or hydrology on branchiodelidans, and branchiodelidans may be equally or more greatly affected by environmental characteristics in host species. So the question is the big water crayfish following the symbionts range and aligning itself with where the symbiosis is occurring, or is the worm nested within the big water crayfish's range and the big waters just like don't care, and it's just more of like a benefit if they're there. And I guess that's to mention as well that these guys can be both commensalists, parasites, and mutualists. It just depends on what's going on. So what I looked at was 3 kind of like sites or groupings. So we have Cranberry Lake Biological Station in green, Newcomb, ESF Newcomb campus in blue, and then like Syracuse sites in red. So thank you Dr. Arsenault and Dr. Cece McNulty for letting me go up there into the Adirondack field stations, the town of Marcellus for letting me sample there, and Dr. John Stella for allowing me to be in his backyard at all, like many hours of the night. And I mentioned 3 abiotic parameters: temperature, electroconductivity, and pH, and 4 biotic parameters of crayfish, so total length, sex, population abundance, and sympatric species. And I use iNaturalist to kind of figure out where they all are in the state. So if you see mild, like 46, identifying your crayfish on iNaturalist, it's me, it's probably a correct ID. And so there's already some differences in sites just based off species assemblages. So you see the cranberry sites are mostly just big waters, while other sites like Abiotic and Newcomb have big waters, brooks, calicos, and clear waters. That's like the highest diversity of crayfish species I've ever seen anywhere in New York, which I was just like surprised that I was able to find that like 30 meters of stream reach. They're also mostly blue, which is also really weird, but that's getting sidetracked. So I measured 30 big waters per site, collected all of their worms, and then in the lab selected the first 15 crayfish I got, and used 2 worms from each for genetics to get 30 worms for each site. And used PBS, basic strategy protocol, which basically we like took the worms and smashed them to bits in a little Eppendorf tube and then threw it through PCR and then just kind of hoped that it worked. And then made a phylogenetic tree from that. So these are the results I've analyzed so far. I will add the caveat that I am very new to R. And so if you have any ideas of analyses or packages that I should be using, please tell me. Right off the bat, this is the worm abundance right here on the y-axis, and then total length on the x-axis. These are all the different sites that I've sampled. You can see that the main thing here is that crayfish worm abundance increases with crayfish size, which makes sense because more surface area, more space for worms. But what I wanted to point out with this is that a lot of the crayfish worms show up around the 60 millimeter mark, which correlates roughly to the 3rd summer year crayfishes, which starts around 60 millimeter, say 60 millimeters, I mean total length, so from rostrum to tail. unfortunately, that means all of my other work over here I'm ignoring for the rest of these analyses. And so even though I have 199 crayfish total from the third summer, I guess, age class from across all the sites, I have not yet managed to get through every single one of them because, for example, Marcellus, which you'll see I have 4 dots right now, takes 2 hours for every crayfish I need to go through to rip off these worms and sort through them. So there's a lot of time that's invested in just this amount as well. So temperature did seem to matter, explaining 24% of the variation. And then for a couple of these graphs as well, you'll notice that the Syracuse sites, so Marcellus and Meadowbrook with SYR next to them, seem to be driving a lot of the relationship. And so I also pulled out just the Adirondack sites, so the Newcomb, the NC, and the CLBS, the Cranberry Lake Biostation sites, and put them in the bottom right corner if you were curious. So temperature seemed to be exponential. I haven't figured out how to do that in R yet, so it will probably be a greater effect once I figure out exponentials. pH also seemed to matter, even though it looks like Marcellus is driving that completely. Yeah. And then total length within the third summer explained a lot of the variation, which I have some thoughts of when I'll get to later, but less in the Adirondack, which was really interesting. And then also these little bands here are 95% confidence intervals. I don't know if I mentioned that yet. By sex, it did matter, but not very much, which is interesting, unless you're in the Adirondacks. In the Adirondacks, it seems to matter a lot, and I don't really know why yet, so if anyone has any ideas why that could possibly be, looking at you, Dr. Reid, I'm not really sure yet. I'm still trying to figure that out. And then my population abundance and electroconnectivity analysis graphs are still under construction, so I'm still working on that. And then, so what species did I find? So I found, this is a picture that I took on the microscope, Cameron-Cola philadelphicus in air quotes, because you'll see it pop up many different times in the phylogenetic tree. And then, as well as Cameron-Cola phallax, which is known to be like a symbiosis with crayfish growth rates. So in heavy sedimented streams or places where there's crayfish who are undergoing heavy gill fouling, these worms will actually eat detritus from the gills and the surrounding body and increase crayfish growth rates in those ecosystems. And so that's one of the species that can be a symbiotic species with crayfish. And then I also found Badilladrilus illuminatus, which is a gill specialist. They're super easy to identify because they look like little grub versus, say, this guy who looks just like a regular worm. So they have more like a grub fat appearance. I also found Xeronogoton instabilis, which is the tennis rackets, and you can kind of see why they look like tennis rackets up close, or why I call them tennis rackets up close. So what? And this is CLBS Sucker Brook, if you guys are curious. So host sex matters, maybe, or I guess it does matter, but not really a lot. It could be possibly because of more agonistic behavior by males, so they fight more, they gain more interactions on a crayfish to crayfish level, and these worms are spreading primarily through direct contact with crayfish. Crayfish will actually actively go out and look for more worms on other crayfish if they feel that the host or the water quality is not like that great. They'll even use, like, there's a paper that says they'll align themselves with the electromagnetic field of the Earth on the north, northeast, and south, southwest axis, and the ones that have too many crayfish worms won't align themselves on that axis. So there's like a really weird, like, I guess, mechanism here where the crayfish are actively seeking them out, but also not wanting too many, and the worms are actively seeking them out, but will also become parasitic when there's too many of them on them, or on the crayfish. So that we could potentially explain behavior by males. They just are contracting more. Host length matters. Obviously, older crayfish will have more surface area, so that makes sense. But it also could be a form of like community assembly and succession. So for example, the Xeronogoton instabilis, which is tennis rackets, which is this top left one here, that would be more of an R-selected worm, and so they may be able to colonize these younger crayfish faster because the younger crayfish are molting more frequently. And the older crayfish will probably most likely have more Cameron-Cola philadelphicus, which is like more case-selected species. It's bigger, it's more competitive, and yeah. So it could be that the older crayfish just have a more stable habitat and they're not shedding off their exoskeleton, which is acting as a disturbance, shedding off more worms. temperature matters. As I said, there is very little papers on this, and so the discussion is hard when no one has done organismal level explanations on these guys. So the only thing I can really find about what could be causing temperature is that it's difficult for them to attach a substrate at 78 degrees Celsius. So it kind of makes sense that the inverse of going down would increase as you go up to a certain extent. What that maximum is for the species in this PowerPoint, that hasn't really been found yet. pH matters. I'm not really sure why. I think it could be potentially a confounding factor where it's marking something else or is associated with something else, and that's being tracked by pH itself. The only thing I can think of is that maybe they are feeding on more seed shrimp in the more basic water. So that's a seed shrimp. Crayfish also have symbiotic seed shrimp on them, if you didn't know. But again, something that I was kind of going here to kind of ask for ideas for about what pH could possibly be doing with these soft-bodied annelids. And the bottom line for this is that it's a super dynamic system. It's not just like a basic model of symbiosis. It's an example of how symbiosis can switch between mutualism to parasitism to commensalism and back and forth all the time. There is so many avenues for research with these guys and exploring more complex systems than some of these other symbiosis systems that you see in like these biology textbooks. Because, for example, we don't know if they're semiparous. We don't know how they overwinter. We don't know what their complete life history is. We don't know many more environmental tolerances for them. And by many more, I mean anything other than pH and temperature. So like literally anything else. And overall, understanding this symbiosis can not only help you understand how the invasion frontiers work with invasive crayfish, because I think I mentioned it before that the invasive crayfish can decrease the diversity and abundance on native crayfish, so it could be disrupting symbiosis. Understanding how, these shifts between symbiosis and mutualism, commensalism, parasitism actually work, and how fine scale it is. Maybe it's site by site, maybe it's day by day, maybe it's season by season. And just overall could be a really good model system because you can culture them in the lab. You just have to have a living crayfish to make more of them. They could just like wiggle around in a petri dish, and they do this really weird behavior when they can smell a crayfish nearby, and they like search like a tick in like circles. So it's pretty cool. Okay. And then what's left? I want to process more samples to increase my sample size for all these graphs. I want to analyze more variables that I have data for, and finish the genetics for each of my sites. So far, I think I have around like 140 sequences about for these guys. And then I want to write my thesis. So thank you for listening. I guess my acknowledgements would be like thank you to professors and people who let me sample on their properties. So yeah, thank you.